Good day all. The presentation that I will be sharing with you today is about a passive sampling device known as the artificial mussel or the AM. This was tested in both a laboratory and a field experiment as a monitoring tool for platinum exposure in the freshwater environment. It has been seen in the environment that pollutants can be found at different concentrations or fraction within an aquatic ecosystem, and this can be influenced by a variety of factors such as pH, conductivity, other pollutants, season, and so many more. Not only is it important to know what is in the water and sediment, it is also important to know how these pollutants affect organisms that are found in these environments and how they accumulate and regulate this exposure. Biological indicators are frequently used for environmental monitoring of metals, since they can give time-integrated results and they are an indication of the bioaccessible fraction of metals in the environment. However, the bioaccumulation of these organisms are affected by various factors such as body weight, growth rate, age, reproduction, and the list goes on. To overcome some of the challenges that bioindicators bring, passive sampling devices were developed and are still being developed. In this study, we focus on the artificial mussel. Like most other devices, the AM works on the principle that the analytes freely flow from the sampling medium into a receiving phase in the device. With the AM, the metals diffuse through a semi-permeable membrane into a receiving phase, which contains Scheele's 100 resin. This is a chelating resin that is used to quantify metals in the sampling medium. The aim allows the determination of the dissolved metal fraction in water bodies without having to kill organisms, as well as environments with unfavorable conditions for living bioindicators. The aim has been used to monitor a wide range of metals in both freshwater and marine environments, and several tweaks have been made to the original design of the aim. So before the study was conducted, the uptake kinetics of the Scheele's 100 resin was first investigated, and it was found that the resin were capable of accumulating high concentrations of platinum. Two modifications were made to the original method, and all the details regarding this can be found in the article mentioned below. The present study aimed to determine the concentration-dependent platinum uptake kinetics of the AM in freshwater over a wide exposure concentration range under laboratory conditions, to validate the AM under field conditions as a reliable tool for monitoring platinum concentrations in freshwater environments by comparing the platinum uptake of the AM with that of living organisms, and then to comment on the appropriateness of the AM as an alternative to living organisms as biological indicators of metal exposure. To validate the platinum uptake by the AM, a static 6-week exposure study with a series of increasing dissolved platinum concentrations were performed. Water and AM samples were collected weekly. During this exposure, platinum uptake was found in all of the platinum-exposed AMs. However, at the lowest concentration, the platinum measured in the AMs was only detected after 6 weeks of exposure. In all higher exposed groups, the platinum increased already after the first week of exposure. In the highest exposure group, the platinum concentrations in the AMs increased over the whole 6-week exposure period, and a steady-state condition were not reached, indicating that the binding capacity of the 200 mg Scheele's 100 resin was not reached. The analyzed platinum concentration in the tank water and the uptake by the AMs were positively correlated, which indicated a concentration-dependent platinum uptake kinetics of the AM over a wide exposure concentration range. This relationship is important when using the AM for monitoring purposes, as it makes it possible to draw conclusions about the environmental platinum contamination, or more precisely, the dissolved bioaccessible platinum concentrations in the water phase. In addition to the laboratory investigations, field sampling of living organisms were combined with active monitoring using AMs at 2 impoundments in the same catchment. The 2 impoundments that were studied are found in the Hex River catchment in a platinum mining area of South Africa. In Olifants Neck Dam, the platinum exposure only originates from geogenic sources, while Bosper Dam is downstream of various anthropogenic activities, such as intensive platinum mining and refining activities, as well as industrial and urban effluents. The main purpose of the field survey was to determine the efficiency of the AMs to accumulate platinum under field conditions in comparison to living test organisms. The AMs took up platinum at both sampling sites. However, Platinum concentrations of the AMs from the two impoundments were identical, even though the two selected sampling sites for this fieldwork differed significantly in the water and the sediment. Bosper Dam showed higher platinum concentration in the water than Olifantsnek Dam, whereas the sediment demonstrated an opposite pattern. This could be explained by the different sources of platinum in the two impoundments. Olifantsnek Dam is located upstream of the mining activities, so that the platinum concentrations are only of geological origin, explaining the higher concentrations in the sediment. In contrast, Bosper Dam collects all the runoff and wastewater from the platinum mines, explaining the higher platinum concentrations in the water as compared with Olifantsnek Dam. However, as the AMs are only able to take up the dissolved metals, it would have been expected that higher platinum accumulation in the AMs would have occurred in Bosper Dam. However, this was not the case. During the 6-week exposure of active monitoring, freshwater clams were transplanted alongside the AMs. During this period, it could be seen that the AMs accumulated identical platinum concentrations, while the platinum in the transplanted clams exposed at the same sampling sites were below detection limit and not illustrated on this graph. In contrast, the naturally occurring organisms, which include different fish species, show to a degree other patterns. These species include the sharp-toothed catfish, Clarias grypinus, the common carp, Cyprinus carpio, and the Mozambican tilapia, Oreochromis mozambicus. The platinum concentrations at Bosper Dam were significantly higher in the fish tissues as compared with the AMs. Two of the three fish species, the common carp and the sharp-toothed catfish, showed higher platinum concentration in the muscle and the liver at Bosper Dam as compared with Olifantsnek Dam, and thus the same pattern as the aqueous platinum concentration. There are significant differences between the metal uptake mechanisms of the AMs and the fish. These three fish species occupy different trophic levels and utilize different feeding strategies. The common carp and the sharp-toothed catfish are both omnivores, whereas the tilapia is an algivore and a detritivore. With the AMs, biomagnification, or the metal fraction that is bioaccumulated via food, is not taken into account. Additionally, the AMs are fixed at one point, while the fish can move around the impoundment, integrating the platinum concentrations over a wider area. The AMs were deployed in the impoundments for a period of 6 weeks, while the fish had been exposed, depending on their age at capture, for a couple of months to a couple of years. It has been thought that the common carp and sharp-toothed catfish specimens were more matured compared to the immature tilapia specimens, which can attribute to a longer exposure period. These differences in metal uptake routes and exposure period can account for the differences in the platinum bioaccumulation between the AMs and the fish. At Bosper Dam, the water hyacinth, Eichhornia crassipes, was also assessed, as this plant is known to accumulate platinum. This was not part of the original sampling plan. However, a hyacinth bloom coincided with the AM deployment period. This photo was taken at the beginning of the 6-week exposure period at Bosper Dam. On any given day, there are hyacinths present in the impoundment close to the banks. However, at the end of the 6-week exposure period, there were almost no surface area that were not covered with hyacinths. Since the water hyacinth is a floating macrophyte and there is no contact between the roots and sediments, it was thought that it would be interesting to see how the metal uptake would compare with that of the AM, since they both can only take up platinum via the water phase. The results showed that the platinum was taken up with the highest concentrations in the roots. This supports other laboratory-based findings, which demonstrates that the water hyacinth is an effective bioaccumulator of platinum. It can be assumed that the high biomass of the hyacinths accumulated metals from the water column to such a high degree, and thus lowered the exposure concentration and metal accumulation in the AMs. So far, the AM has only been validated for 4 metals in the marine environment. In the present study, the use of the AM was adapted for the monitoring of platinum under freshwater conditions. The field exposure demonstrated that the AM is a promising tool for monitoring platinum in the freshwater environment, and the laboratory study demonstrated that it accumulates platinum in a concentration-dependent manner. The AM can only present represent the dissolved bioavailable metal fraction and does not consider particle-bound metals, which can be taken up by living organisms via the food. Nevertheless, a significant advantage of the AM is that its use is largely standardized, so that many influencing parameters can be excluded. It should be noted that the differences between the AMs and other test organisms would also occur if different species of organisms should be used and compared with one another. Thus, it is recommended to combine active monitoring using living bioindicators with AMs to obtain insight into the role of dissolved bioavailable metal fractions and particular bound metals in metal exposure in aquatic ecosystems. As there is a big constraint for presentations, a lot of the finer detail regarding methods, analyses, and discussion on these results could not be mentioned in this presentation. However, extra information can be found in the below mentioned literature. Thank you very much.